Satyam Ideal Learning. And guys, our today's topic is about artificial vegetative propagation. So we will be discussing about artificial vegetative propagation. We have already discussed about natural vegetative propagation. Now you will say, we have not discussed natural vegetative propagation because vegetative propagation that I have told you that there are forms by which it is growing. The plant is growing from underground stems, the plant is growing from subaerial stems, from it is growing from roots, it is growing from leaves. These are the only things which comes in natural vegetative propagation. So guys, these things that I have already taught you, this comes in natural vegetative propagation, and now we will be discussing about artificial vegetative propagation. Now, what comes in artificial vegetative propagation? At the very first, I have told you one thing, that the new plant which comes, which is produced from the vegetative propagation attains maturity earlier than that of the new plant which is produced from seeds. Okay, so this thing I have already taught you. Now, in artificial vegetative propagation, cutting. Artificial vegetative propagation is of four kinds, is mainly of four kinds. One is cutting, one is layering, one is grafting, and one is tissue culture, or it is also known as micropropagation. Tissue culture is also known as micropropagation. We will be discussing all about them. Okay, so watch the video till end. So let's start. What happens in cutting process? So it is a kind of artificial vegetative propagation in which the stem of the plant is cut, and then the cut end is taken and placed in the moist soil. We will place it in the moist soil. Like this is a stem and we have cut this portion. Now it has been cut. Now this plant we will take and we will place in the moist soil. Now it will, now the roots will come after some days. Now you have placed this cut end in the moist soil. Now what will happen after few days? The adventitious roots will be coming. Okay, so all the roots will be coming. This is how now the plant will grow individually. Okay, so the plant will form a new plant. Okay, now what are the examples? The most common example is of rose. Okay, rose. You already have been knowing that in a rose we cut a stem and then we will separate all the leaves together, and then we will take the stem, only the stem that should have buds, and then we will place it on the moist soil. Then it will grow after few days when we will watering. Okay, we will watering also the plant regularly. Now come on, layering. Now layering, what is layering? So I am rubbing this thing. Now in the layering process, what happens? See, like this is a tree, like this is a tree, and these are the branches. I am making very roughly diagram. These are the branches. Now what we have to do, we have to take one of the lowest branch, the branch which is very close. So we will take one of the lowest branch, like this is a branch which is very close, and then I will take the branch, and what I will do, I will bend it downward. Okay, so I will bend it downward. Now this is the soil. If I bend this downward, it will come like this way. See, if I bend this downward, then it will be joining with the soil. Okay, then it will come in the contact of soil. Now what I will do, I will take the lowest branch and I will bend it down, and I will try to touch it with the soil, and then what I will do, the portion which is touching the soil, we will be covering that portion. Okay, we will be covering that portion with the soil, the last portion, this portion, we will be covering with the soil. Okay. Before covering with the soil, we will remove the ring of bark. Okay, we will remove a ring of bark before covering it with the soil. So what we will do? We will take the lowest branch of the tree and then we will bend it downward and we will try to touch it with the soil. And when it touches with the soil, then what we will do? First, we will remove the portion that we have to cover with the soil. So we will remove the bark of that portion. Okay, we will remove the bark of that portion which we have to cover with the soil. Now what we will do? We will cover it with the soil, the portion which we have removed the bark. Now what we will do? We will place a heavy object. We will place a heavy object like this is a heavy object which I have placed. And now why I have placed this heavy object? So that it will be in place. It will be in place because the root, the branch is as flexible so that it can come upward. So it is as flexible. No. So for keeping it in the place, we will take a heavy object and put on it. Okay, now what will happen? After some days, you will keep on watering regularly. Then what will happen? There will be some adventitious roots will be arising. And now what we will do? We will cut the main branch. We will cut the main branch. And the plant will go independently as a new plant. Clear? This thing is about one kind of layering. Okay, so it is the one kind of layering. There is another kind of layering also. Now in that what happens? We will take a branch. This the examples are of this kind of layering. The examples are rows. Okay. Now we will take a tree like or a plant you can take. Now I will take one of the branch of the plant. Now okay, of a tree or anything. We will take the branch. One of the branch we will take. And now what we will do, we will cut the bark portion. Okay, we will remove the bark portion like this way. If I have to use this portion, okay. So what I will do, I will remove the bark of this much portion. Okay, of this much portion, I will remove the thin layer of bark. And then what I will do, I will cover it with the soil and then I will wrap it with a rag or a straw. Okay, so with a straw or a rag you can wrap it. Okay, now what you will do, you will water it regularly. Okay, you will water it regularly and then what will happen, there will be arising some adventitious roots. So the adventitious roots will be arising like this way. And then what we will do? We will separate it out and then we will take it and we will grow it in the moist soil. Clear? Now what we will do? We will take it and we will grow in the moist soil. Okay? And this will grow into an independent plant. I think the layering process is clear. Okay? So after a few days it happens. I think it is very clear. And the example of this kind of layering is, I think it is known as air layering. Okay. These examples are mango. The example is mango. Now, we come on grafting. Now, what happens in grafting? We have to take two parts. We have to take two parts. One is scion. One is scion and one is stalk. So we have to take two parts. One is scion and one is stalk. Now what is scion and what is stalk? Scion is a young cut part. Okay, it is a young cut part of a stem. Like if there is a plant, if this is a plant, I have cut the young part of that stem. Like this is a stem which is grown and this is a young stem. So I will cut it and I will take it for breeding. In grafting process, no, breeding occurs. We take a stalk and we take a scion, and then we will breed it out. Now how we will breed it out? Listen it carefully and then you will understand. So it is a young cut portion of a plant. Okay. So scion is the young cut portion of a plant. Now what is stalk? Stalk is already a base of the stem, which is already been already has roots also. It is already in the ground. Okay. So like if this is a ground and this is stem which is there. So this is the stalk. This is the stalk. Okay. So it is already been rooted. You are seeing in the land it is already been rooted. So stalk is already been rooted, and scion we will take it from any other species of the same species. Okay. We will take it from the same species or of the related species of the stalk. Okay. So we will take the young piece, and what you will do? You will take it and you will put it, and then you will take a kind of cloth and you will wrap it out tightly, very tightly you have to wrap it. Like see, this is a scion, and now what I will do, this is my stalk. Now what I will do, I will take this scion to here and I will wrap it out very tightly. I will wrap it out with the cloth. Okay, outside से wrap कर देंगे हम. Okay, we'll wrap it out very tightly. And now what happens? It will become a single plant and grow. After few days or a month, it will grow as a single plant. Okay, and in this kind of thing, we can breed out also. Like if we want that in the mango plant, I want that the color of the mango should pink. Not pink, sorry. We can change the sweet color. Sorry guys. We can change the taste also. We can change the color also by doing this kind of grafting. Okay. By doing grafting, we can change the color or the taste of a plant. So, if there is a mango plant which is already been rooted, I can change the color of the mango plant by taking some related species and put it on this as a scion and then wrap it out, and then it will grow as a single plant. There are three types of tongue grafting, crown grafting and bud grafting. Okay, and there is one more, wedge grafting. So I will telling you all wedge grafting. Wedge grafting is there, tongue grafting is there, crown grafting is there, and bud grafting is there. Now what happens in tongue grafting? Same procedure is there. Only the thing is that we have to take a stalk and a scion. Like this is my stalk, and I have take a scion. Now what we have to do now? Like this is my scion. Stalk. This is my stalk, and what I will do? I will make a mark. I will make a cut mark like this way. If I make like this way a cut mark. Now my scion should have an opposite kind of cut mark so that it can fix up. So my scion will be having like this way cut. And then it fixes on. And then what I will do? I will wrap it out with the cloth. Okay. I will wrap it out and then it will grow as a single plant. This is a kind of grafting which is known as tongue grafting. Okay, this is a kind of grafting which is known as tongue grafting. Now in crown grafting, what happens? First I will tell you wedge grafting. Wedge grafting, W is formed by two V and V, which forms a W. Now what you have to do? You have to take a scion and a stalk. Now stalk is this and the scion is this. Now you have to make a mark V, and now you have to put it on. When you will put it, then it will come like this way. See, I will make you very clearly. then it will be like this way. And now what you have to do? It has been fixed on, no? Are you seeing, guys? It has been fixed on to the stalk. So the scion has been fixed on to the stalk. So this is scion and this is the stalk. It has been fixed on, and now what we will do? We will wrap it out with a cloth. This is the wedge grafting. Now in what happens in crown grafting? In crown grafting, many scions are added to a single stalk. The diameter of the scion will be smaller than that of the stalk in kind of crown grafting. Okay, in kind of crown grafting. In tongue grafting and in wedge grafting, the diameter of the stalk and the scion will be same, and the scion should be of the same species of the stalk or should be of the related species of the stalk. Clear this thing? Now what happens in crown grafting? I think this much you have been clear very nicely. Now what happens in crown grafting, guys? We will take a stalk. Now what you will do, like this way, it is the stalk. You will make a mark like this way in the sides. Like I am growing 3 scion. I am growing 3 scion. Then what I will do? I will make 3 mark like this way. And I will take 3 scions which have the same marking, which can fixed on. Like this is a scion, one kind of scion, and it has been fixed on. The diameter should be changed. Now what you will do? You will wrap it out with a piece of cloth. Okay, you will wrap it out very carefully with a piece of cloth, and then you will leave it, and then it will grow as a single plant. Okay, with more scion. Clear this thing, guys? Now in bud grafting, what happens in bud grafting? We will take a stalk. So this is our stalk, guys. We will take a stalk, and what we will do? We will make a T-shaped cut. We will make a T-shaped cut. Now if we make a T-shaped cut, like this, I have made a T-shaped cut. Now when we will try to open this, now this has been opened like this way. Okay, it has been opened like this way when we try to open this. And now what we will do, we will take a bud and insert it in between. In between we will take a bud and insert in, and then we will close it, and then we will take a piece of cloth, and we will tie it out, and then we will leave it alone, and then we will water it, and then it will grow into a nice plant. First of all, we have to remove the bud which is already growing, so that the bud which we are trying to grow, it can grow up. Okay, and one thing, it is more important, scion is also known as graft. Scion is also called graft. Okay, so first we will remove the bud which is already there, and then we will insert the bud which we have to graft. Okay, which we have to graft. So bud is the scion, and this is the kind of stalk. Clear this thing about grafting? Now we will be coming on tissue culture. As I have told you already, that tissue culture is also known as micropropagation. So what is known as tissue culture? Tissue culture is also known as micropropagation. Now see, the tissues of a plant, if there is a plant and it has tissues, okay, it is made up of many kind of tissues, no? So in the tissue of the plant, in the single cell of that tissue, in the leaf also, in the stem also, it has the inherent ability to grow into a new plant. Even a single cell of a plant tissue has the inherent ability to grow into a new plant. Okay, so A single cell of a plant tissue has an inherent ability to grow into a new plant. Now, tissue means, what is the meaning of tissue? The meaning of tissue is that tissue is made up of similar kind of cells which have the similar function. So, the cell which have the similar structure as well as the similar function forms the tissue. Now, what is the meaning of culture? In Hindi we say growth karana. Okay. Tissue ko growth karana. This is tissue and we are culturing the tissue. So, we are making the tissue growth. Okay. So, culture means growth karana. Okay. To make grow. So, tissue culture meaning you have already been understand. So, all the plant tissue and the single cell of a tissue has the inherent ability so that it can grow into a full plant. Clear this thing? And tissue culture is happened in medium. Okay. So, we will take a tissue, we will take a plant tissue and we will culture it. Means we will make the tissue to grow on a growth medium. Growth medium is a substance. Growth medium contains certain types of salts. It contains certain types of plant hormones like and it contains certain types of antibiotics. So growth medium is made up of, it contains like antibiotics, growth hormones of the plants. Okay, so growth medium is that thing. And what we will do, we take the tissue and we will make the tissue culture. We will make the tissue to grow on the growth medium. Okay, so we will take a bowl and in that it is growth medium and we have placed the tissue is there. Tissue is there and then we are trying to grow. Now, what is explant? What is explant? Explant is the plant tissue that we will be culturing, that we will be making to grow. Okay, so the plant tissue which we are trying to make it culture, to make it grow, that is known as explant. Are you getting my point? Now what we will doing, we will take the explant and we will sterilize it. We will make it germ free. And tissue culture happens in a germ-free environment, in a sterile environment in the laboratory. Okay, so we will take the explant and we will make it germ-free by sterilizing it. And then what we will do, we will take the explant, the cell of the explant, and we will put it in the growth medium. Growth medium contains a hormone. There are two kinds of hormone that I will be telling. These are the plant hormones, auxin and cytokinin. Auxin is a kind of plant hormone which promotes the growth of root, and the cytokinin hormone, the cytokinin is also a plant hormone which promotes the growth of shoot. Okay, so it promotes the growth of shoot and it promotes the growth of it promotes the growth of root. Okay, so these are the two. So this promotes the growth of root. So auxin promotes the growth of root and cytokinin promotes the growth of shoot. Clear this thing? Now we will take the explant and we will sterilize it and we will put it in the growth medium. Growth medium means it has hormone like auxin and cytokinin. And then what will happen? It will grow. This tissue, this cell will grow. Okay. And it will divide and redivide and forms a mass of undifferentiated cells. Okay. So it will form a mass of undifferentiated cells. Okay. So undifferentiated cells. It will form a mass of undifferentiated cells. Undifferentiated cells are those cells which are not yet a part of a kind of tissue. Okay, they are not yet a part of tissue. Okay, And this undifferentiated cell, a mass of undifferentiated cell is known as callus. So callus is a kind of mass of undifferentiated cells which has been formed by a single cell. Okay. And then what will be there? We will take the different kind of growth medium and we will make the tissue to grow in different way, in the different way of shoot and in the different way of root. Okay, so we will take the different kind of growth medium and we will make the plant to grow from in the different way. Like if a shoot has to grow, how the shoot will grow, how the root will grow, in the different manner it will grow, na? So we will take the different kind of medium, growth medium, and we will make the tissue to grow in it. Okay. Now what will be done? All things are done. Now we will take the plantlet which has been formed and we will grow it as a single plant, as an independent plant. And in tissue culture there are many variety of plant, means not a variety, means there are many plants which are grown. Okay, it's a kind of vegetative propagation, artificial vegetative propagation, in which a large number of plants are grown. Okay, so there are there is a variety of large number of plants which are germ free, disease free. Okay, so the plant are germ free. This is the advantage of the tissue culture. So the plants are germ free. So if a parent plant is having any kind of virus, then it can be so the baby plant, or you can say the new plant, will not have that virus. It will become a disease free or a sterile or a germ free plant. Okay, so this is the advantage of tissue culture. So bye bye guys, we will meet the next time very soon. Thank you.